There we go. All right, let's start. So I think this time we'll just go through the questions sequentially. So let's start at the beginning. Have a read of this. Okay. Might be easier if I just pass you this.
Okay.
Yep, that's right. So tell me how you know that's the cell in anaphase.
Because you can see the sister chromatids being pulled to opposite sides of the pill.
Good.
The cell.
And if I was to ask you to show me cells that are in the other phases, could you do that?
Maybe. Okay. So I think this one is in the prophase, the early prophase.
Okay, so talk me through that. What's happening?
So there's chromosomes that are condensing. Yeah.
And what's happening to the nuclear envelope?
It's vanishing, or it's being broken down.
Yeah, good. No, vanishing. So what's happening later prophase then?
So in the later prophase, the nucleosome is broken down, the envelope is broken down, the chromosome are finished coiling, so they're condensed, and the centrosomes are starting to move towards the opposite poles of the cell.
Yeah, good. Okay. And metaphase?
In metaphase, the chromosomes are being aligned at the equator.
Can you see any of them here?
Could be this, but
No,
not really.
there aren't any. Good. And there's anaphase there. How about telophase?
Oh, okay. So telophase is where the chromosomes uncondense and the nucleosome synthesized together with the envelope, and yeah.
And what happens after telophase?
Cell division.
Yeah. Which is called?
Cytokinesis.
Good. Perfect. All right. Full marks. Let's move on. So next question.
It's just the advantages of using a light microscope rather than an electron microscope. Okay, so I don't really know, but I'm gonna guess.
Okay, what would you guess?
Okay, so the electron microscope can give us too much detail into the cell that we don't need for cell division.
I mean, if you're studying cell division, detail is good. If you
Yeah.
want to study it, detail can only help. Do you know how we prepare specimens for light microscope?
Okay. Yeah. So in light microscopes, you can use a live tissue, so it doesn't have to be dead. And in electron microscope, there isn't supposed to be any air or water in it, so it's hard to produce a lifelike specimen of like the cell or whatever.
And why is that important for this?
Because cell division is part of a cell cycle, so it has to be cell that's going through. Something like that.
Precisely. Okay. So 2 marks there. One of them is from the fact that you can actually see living cells with a light microscope, and the other one is that therefore you can see the cells going through mitosis, basically.
Yeah.
You could pick up the second mark by adding other details, like saying that light microscopy is easier to use. You need less specialist knowledge and expertise to use a light microscope. Or you could talk more about the preparation. So you could say that in light microscopy, you don't need to use heavy metals, therefore it doesn't kill the sample.
Mm-hmm.
And it's less time consuming as well.
Mm-hmm.
But yeah. Good, next one.
Okay, so for repairing tissue,
Yeah.
For asexual reproduction,
Yeah.
for growth, oh no, growth is only, for I don't.
Okay.
Yeah.
I mean,
oh no, for stem cells.
Okay, what do you mean about that?
So stem cells use mitosis to create cells which then differentiate into specialized cells.
Yeah, you definitely get the mark for that. The other points that they use, so when you're talking about repairing tissue,
Yeah.
you can make that 1 mark into 2 marks by saying repairing damaged tissues and replacing dead cells.
Okay.
But otherwise, you're right. You pick up a mark for asexual reproduction, and I mean they also say if you say production of genetically identical cells, you pick up a mark. But that's essentially just explaining what mitosis is. So that's not necessarily how I would have answered the question, but I think you're on the right lines. Just remember when you're talking about repair, specify repairing damaged cells and replacing dead cells.
Yeah.
Okay. Next one.
Okay. Yeah. Okay, so I don't think it's an artery. I think it's a vein because it has a large lumen.
How large is the lumen?
Oh wait, there's a it's a capillary.
Good.
Yeah, I didn't see the
scale.
The scale. Yeah.
Fair enough. They could still ask you that question without a scale bar because you were told that it's an electron micrograph.
Yeah.
Electron micrographs are very high resolution.
Okay.
Tell me what else you see there. Now that you know that it's capillary, what do you know about capillary that you could identify in that picture?
You could identify the tissue fluid surrounding the capillary.
Yeah.
You could identify the epithelial lining.
Endothelial lining.
Endothelial lining of the capillary.
Tell me about the endothelial lining of the capillary.
It consists of a single cell layer.
Okay, so point to the cell.
Yeah, there.
Yeah, where's the nucleus?
Like there.
So this whole thing here is the nucleus. I mean, it's fair, like, I know that you probably haven't seen many images like this, but this would be the nucleus.
Okay.
And then the cytoplasm is literally forming the wall, the lumen. Now, when you said it was a capillary, you're right, so you get a point for that. And if you were to say, like, reasons for your choice, could you tell me now why, from looking at this, you could say this is a capillary? What made you think that it was a capillary after you saw the scale bar?
So I know that the diameter of the capillary is equal to 7 micrometers.
Good, that's 1 point.
So, also because the thin wall of the vessel.
Yeah, you'd get the mark for that, but I would specify single cell thick.
Okay.
Otherwise, any other points you might be able to relate to capillary?
The environment of the capillary, so it's tissue fluid.
Yeah, I mean, the fact that it's, immediately exposed to the extracellular fluid tells you that it's also capillary, because it's like the cells that it's supplying are literally sub-micron distance from it. So that gives a clue that it's a capillary as well, and you pick the point up if you said that. This one's a bit harder.
The pulmonary artery. Okay. Okay. So it closes where they both intersect.
Good. Which intersection?
So it would be at around 0.25 seconds.
Yeah, you get that mark. 0.24, 0.25.
Okay.
Good. When do the ventricles begin to contract?
At 0.08 seconds.
Good. Yeah, that's right. So start with the similarities.
Okay, so the curve, the shape of the curve would be exactly the same. But everything would be shifted upwards.
Okay.
Because the blood pressure is higher because the left ventricle has to, as part of this systemic circulation, so it pumps blood throughout the entire body, while the right ventricle just pumps it towards the lungs.
Good. That's 3 out of 4 marks.
Okay.
You can pick up your last mark by just going into a bit more detail. So when you say it pumps it to the whole body, why does that mean that the pressure will be higher?
Because it has to reach tissues which are more distant to the heart than the lungs.
Which you could also rephrase by saying it works against a higher resistance because it's working against basically a bigger surface area of vasculature.
Okay, yeah,
And you pick up your 4th mark that way. That was excellent in terms of describing the differences. In terms of describing the similarities, whenever you're like explaining something with reference to a graph. Just be like
Really precise.
Yeah, really precise. So say
Ridiculous.
the rise in pressure would be at the same time point. The fall in pressure would be at the same time point. That kind of thing. You will pick up marks. I know it's ridiculous, but that's what they're assessing. That's what they want to see you can do. But good. That's good. Let's move on.
How's myself?
Good. Next one.
Okay. So antibodies contain antigen receptors of their variable regions. They have a specific shape which allows it to bind to those antigens.
What do you call that? Specificity? So when we say, so it's the same word that we use when we describe base pairing. So C always binds to G, A always binds to T.
So it is a complementary.
Good, yeah,
shape.
exactly. And if you just said that it corresponds or has a matching shape, you wouldn't pick up the mark. They want you to use the term complementary binding site.
Okay.
But good, yeah, you get that. You said variable binding site, that'll give you another point.
And so okay. So I only got 2 points.
Yeah,
Okay.
Keep going.
Okay, so it'll bind to complementary proteins of the cell membrane of the antigen iris.
Good. So what are those antigens they told you in the question?
H or N.
Good. Now, so far you've talked about antibody and how it would bind to either of them, but they've given you 2 different ones, so you can explain how it would bind differently.
So the antibodies which bind to the H won't bind to the N because they're different. They contain different variable regions and different proteins. The virus contains different proteins on the cell surface membrane. So it's not complementary to.
Good. Yeah, perfect. You've seen this 100 times.
Vibrio cholerae.
Good. Next one.
It can be transmitted. Okay, so through. So either it's waterborne or foodborne. So by drinking contaminated water by feces, infected feces getting into the water through bad sewage systems, or by somebody infected with cholera cooking and the bacteria getting into the food and people consuming the infected food. Okay,
good. You would get the marks for that.
Oh, that's too much.
You're giving good answers, but it's lengthy. So you pick up the first mark by just saying fecal oral route.
Okay.
And you pick up the second mark from saying it goes from infected person to uninfected person by contamination of the sewage and then usage of the contaminated sewage by the uninfected person. So I completely understand what you're saying and you would get the marks for it, but just in terms of being efficient, you want to reduce the amount that your examiner has to read. So you're on the right tracks, but whenever it comes to one of these infection questions and you need to describe transmission, like even drawing like headings, uninfected and an arrow infected, like separating the 2 people to show like really precisely how it's being transmitted will get the marks.
Okay. Okay. Lysis being breakdown of the bacterial cell. Okay. I don't think I've really had
That's okay.
this.
Bacteriophages, it's niche knowledge, you wouldn't have necessarily heard about them, but what they're testing here is more the concept of lysis.
So phagocytosis is the lysis in the breakdown.
So lysis is very specific. It means the breaking of the cell's barrier, whatever separates the cell from its external environment. Lysis is when you destroy that and it causes the contents to spill out and the cell essentially bursts. When we talk about bacteria, what is that barrier separating it from the external environment?
The cell wall.
Good. So to pick up the first mark here, you say the cell wall would be damaged, or the cell wall would be broken down.
Okay.
And then you pick up the second mark from either detailing that cross-links of the cell wall could be broken down here. So you could be decreasing the quantity of murin or peptidoglycan.
Yeah.
Or you could say that further to the cell wall breaking down, the cell membrane would also break down. Mm-hmm. Or you could say that potentially the bacteriophage. So when you're thinking about cell lysis, where have you encountered this before, outside of the context of infection? If I gave you a sample of red blood cells and asked you why is that?
Yeah, so when red blood cells take up too much water, or animal cells take up too much water and can't deal with the osmotic pressure.
Yeah, so if this bacteriophage destroyed the cell wall of a bacterium and then somehow also caused more water to be taken on, or because the cell wall is destroyed, bacteria loses its ability to regulate water transport, you can have a huge amount of water entering the bacteria, and that could be what's causing the lysis. Those are the kind of points that they want you to touch on there. Okay, let's do this one and then take a break.
Okay. So bacteriophages would be able to break down the cell wall of Vibrio cholerae before it secretes the toxin. Collagen into the cells of the small intestine.
Okay, so you're focusing on the fact that the bacteriophage is going to kill the cholera, right?
Yeah.
But what have we said? What is a bacteriophage?
It's a virus which can enter the bacterium.
Yep, correct. And if scientists wanted to use that to treat people who are infected with cholera, what is the main concern there?
That they would just infect other cells.
Yeah, which
Human
cells?
cells.
Exactly, human cells. So suggest the properties of bacteriophages that would make this possible. So the bacteriophage would have to specifically infect cholera cells.
Okay.
So that's one of the...
Okay, I was reading the question a bit wrong, I think, then.
That's okay. And it would have to, aside from not infecting human cells, it would also have to prevent side effects.
Yeah.
So you wouldn't want it to just infect cholera cells, but then cause cholera to release all of its toxin anyways.
Mm-hmm.
And you'd get the same problem.
Yeah.
I'll just finish the question then.
Okay. Well, measles is caused by virus.
Good. That's it.
That's all.
That's it. Just gonna
Okay.
say antibiotics are not effective against viruses because this is a virus. All right, let's take a break here. keep going.
Okay. A stop codon is a triplet code when read by RNA polymerase. It stops catalyzing the bond between phosphates and pentose sugars. So it stops forming mRNA.
No.
No? 100% no.
So,
Oh, wow.
What is a codon?
Oh, sorry. Okay, so mRNA codon, it's not a triplet code, so it's a codon. So it's a nucleotide sequence when tRNA binds to it, then it stops forming a polypeptide.
Okay, so what is it? What process is it terminating?
Translation.
Good. There you go. That's the mark. I know it's confusing. But let's scroll back up. Yep. So 4 marks.
Right.
What did it say?
Global structure. 30. Okay. Okay. The ASP and GLN R groups are both hydrophilic. So in globular, proteins they'll be present on the outside in order to make the protein soluble in water.
Good.
And the ILE is hydrophobic. So this R group will be present on the inside with attracted to other hydrophobic R groups in order to keep the globular molecule soluble.
Yeah, okay.
And help its structure.
Good. So explain to me why aspartame and glutamine are hydrophilic and why isoleucine is hydrophilic.
So it contains a carbonyl group, which contains dipoles, and that O can form so it can form hydrogen bonds with water.
Good.
That's all. Yeah. So ASP and GLN can both form hydrogen bonds with water. And the third one can't.
Why can't the third one?
Because it doesn't contain any polar bonds.
So what kind of molecule is it?
Yeah, it's non-polar.
Good. So you've got the marks. When you're describing R groups, make sure you say polar or non-polar, and that's why it has hydrophilic or hydrophobic properties. But in terms of what makes it globular and water soluble, you've got it right. That's perfect. So let's move on.
Glycosidic bond.
Okay. Okay.
Okay.
This is a formal question.
And it's graph. Okay. So okay. The allogenate or the matrix which the enzyme is bound to in the immobilized enzyme makes the enzyme less susceptible to changes in temperature. So enzyme activity is not altered as much as free enzymes, which don't have those molecules bound to them.
Okay, you're right, but you're going into the interpretation now. So what they're asking you to do is just compare the results. You're telling me why the results are different.
Yeah.
So I just want you to compare those two lines.
Okay. So
like very simple stuff here they're asking for.
So the optimum temperature of the mobilized enzyme is 46 Celsius, and the optimum temperature of the free enzyme is 37. Yeah.
That's 2 marks.
Okay. And the enzyme activity at optimum temperature for the free enzyme is 8.3.
2, 3, yeah.
While the enzyme activity at optimum temperature for the immobilized enzyme is 7.6.
Good.
And the gradient, well, no, okay. Is that enough?
That is enough, but go on and explain the gradient for me, because in the exam you wouldn't risk it, you'd write everything.
Okay. So the gradient from temperatures 20 to 30 Celsius. No, no, actually not. Okay. The gradient from 20 to 25 Celsius is bigger for the immobilized enzyme than the free enzyme. The gradient from 25 to 30 Celsius is bigger for the free enzyme than the immobilized enzyme. The gradient from
Okay, yeah, so you could go on and explain all the gradients. I mean, I wouldn't necessarily do that, but when you want to compare what you want to do in each of these cases. So in the previous question that I think we went through last week, and you literally had to do that.
Yeah.
It's almost there's almost not enough to talk about, so you had to talk about that.
Yeah.
But here,
there's a lot.
Yeah, there is more. So you've commented on the optimum temperatures being different, and you've talked about like the maximum activity being different.
Yeah.
So you've talked about the differences. When the question says compare, you can talk about similarities as well.
Okay.
So you could literally say both of them rise to a peak and then fall, and that would get you a mark.
Yeah.
You could say that both of them were active over the range tested, so between 20 and 55 degrees. That would also get you a mark. If you want to like go into the minute, I mean, you would get the marks if you start talking about the gradients, but because it's not that relevant to the experiment, what is relevant is saying that at temperatures below 40 degrees, the free enzyme always had higher activity, whilst at temperatures over 40 degrees, the immobilized temperature is high. But yeah, that's, I mean, so many marks there for only 4 points. So I would just like always comment on optimum temperature, always comment on maximum activity, and then if they're saying compare, you're free to comment on the similarities. So just state the obvious.
Okay.
They both rise and fall. They're both
Yeah.
active with the temperatures tested.
Yeah.
Okay.
Okay. So the Michaelis-Menten constant is the substrate concentration which allows the enzyme, which allows for the rate of reaction to be half that of the maximum velocity.
Good. Come on, that's the mark. I have to mention that Km substrate concentration. You have to mention that it's half of Vmax, but you did that, so good.
So, okay. The Michaelis-Menten constant of the enzyme free in solution is smaller than that of the immobilized one because the enzyme has a higher affinity towards the substrate.
So which one has a higher affinity?
Free in solution.
Yeah.
Which allows Vmax to be reached at a lower concentration.
Good, that's one mark. So here's your
Yeah.
can explain.
Yeah, demobilized. Okay, so the mobilized enzymes contain a matrix or certain molecules which are bound to the enzyme, while those free in solution don't. And those can, by having that, it means that there's collisions can't happen exactly the same way as they can happen with free enzymes.
Okay. In every enzyme question, they want you to talk about a certain part of the enzyme. If you're going to go into explaining mechanisms, what's the most important part of the enzyme?
Active site.
Good. And how could this be different in the free and in the immobilized? So you've touched on
It can be.
the collisions.
It can be partly surrounded by the
Yeah.
immobilized.
So less likely for active site collisions because it's immobilized. Also, what could immobilizing enzyme do to the active site?
Can alter that. So
Yeah, chemically, you can have a structural change in the active site that slightly reduces the affinity. So
okay.
there's your marks.
Apparatus.
Good.
Body.
Good.
Animal, plant. Not bacteria.
Good.
Formula M equals. Wait. Yeah. M equals I. No. M equals I divided by A.
Good. That's one mark.
And then, so you would measure the scale bar.
It's 29 millimeters.
Okay. And then 29 millimeters is 29,000 micrometers. And you divide 29,000 micrometers divided by 2 micrometers, which is 14,500 micrometers, which you could convert into millimeters, but I think they asked for. Okay. Then you've got, and then you've got 14,500.
Good. 4 marks.
No.
Yeah, if you wrote micrometers, you would lose it, but I mean, they're giving you the units, you're not going to make that mistake.
Okay. Let's make constructors. Oh, okay. Chloroplast, mitochondria.
Good. Yeah. Any others?
Nucleus.
Good. Yeah, those are the three. You choose two. Excellent.
Okay. So fluid, because the phospholipids can diffuse throughout the membrane, as well as some of the proteins.
Good. One mark.
Mosaic, because the appearance of the cell membrane when viewed from above. Because the proteins are at different sizes than the phospholipid heads.
Yeah. So amongst the proteins as well, there's variation.
Okay.
So mosaic is, we're just talking about interspersed different proteins on the surface. But yeah, you get the marks.
Okay. So the cell surface membrane creates a barrier with which we can identify the cell as a cell. It creates a barrier to create a certain environment inside the cell where specific chemical reactions and metabolism can occur. So it contains a phospholipid bilayer, which helps the cell to control to a certain degree, or can control the hydrophilic molecules and ions that are exchanged in and out of the cell.
Okay, good. So you're talking still about the sort of barrier aspect of the cell membrane. Yeah. So they're going to give you a mark for focusing on that. When you state this, and it's likely that a question like this will come up in your exam as well. Cell surface membrane is like a favorite. The first thing you want to say is what it's separating. Kind of applies for all barriers. What does the cell membrane separate?
The cell and its environment.
What part of the cell?
The cell wall. The cell, or if it doesn't need to have a cell wall. So it separates the protoplasm and the environment.
Yeah, okay. Protoplasm.
Cytoplasm.
Cytoplasm, yeah. Good. And the external environment. Okay, that's fine. And then you can say something about the violet layer and allowing selectivity and ions that can pass through. Perfect. Now, moving away from like the barrier functions, what else is the membrane important?
Okay, so it contains specific proteins which are part of the signaling pathway.
Good. One more for cell signaling role.
Contains proteins which allow us to identify cells as cells or foreign cells.
Good. So
proteins.
Access cell surface antigen.
It contains endocytosis receptors in order for endocytosis to occur.
Yep.
It contains proteins for the cytoskeleton.
Good. Yeah. Anchoring cytoskeleton.
It contains enzymes.
Yeah, another point. I mean, you're over the top here, but like, this is good. Keep going. Anything else you can think of?
Oh, yeah. It contains cholesterol in animal plant cells. In animal cells, which allow us to regulate the fluidity of the cell.
Yeah, good. Good, perfect. So there were 9 potential marks that you could get there, and you got I think the only one you didn't mention was cell-to-cell adhesion, which is again just another role of the proteins that are going to be on the cell surface. But very good. Next one.
Okay. So the xylem vessel elements are strengthened with lignin.
Okay, so how does that?
It helps the mechanical function of the xylem to be able to handle the negative pressure which occurs due to transpiration.
Good.
Okay. Contains a small diameter in order for air bubbles not to. Actually, I won't take that one. That's a bit weird. Okay. Let me think. Okay. It contains a hollow lumen, the xylem vessels.
Yeah.
In order for minimal resistance against the water and mineral flow to the vessels.
So what does that give you then, in terms of flow?
A higher rate of transpiration.
Good. You get the marks there?
What else was that?
Okay, so
This one.
no. Fair. So you went straight into talking about lignified walls and the lack of cytoplasm, which is that's really good. That's like a bit deeper knowledge than what's necessary. But they're in the marking scheme. You'd get the marks for that. The marks they say are just mentioning that they have cellulose. That would be one description of xylem vessels, and that allows adhesion of water.
Yeah, because it's okay.
Bonding. Another one that you can go for is that the cellulose wall is thick, which prevents collapse under tension.
Mm-hmm.
You talked about lignin. Other than the function that you mentioned of having, it also it has like a waterproofing effect.
Okay.
To prevent water loss.
Yeah. You can talk about pits.
You talk about pits? Yeah, go on, tell me about pits.
So they're regions of the xylem vessel without lignin, which allows water to move out of the xylem.
Good, yeah, supply water to the surrounding cells. End walls of xylem? Tell me about that.
There are no end walls
Good.
of the xylem vessels.
And yeah, that's again ease of flow. And the lumen size.
So that's what I was talking about. Is
Small or large?
Small.
No.
Large.
Right. If you think about like the function of xylem is just to move as much water as possible around the plant, then it's kind of used to extract large lumen to transport a lot of water. I mean, in practical terms, it's not necessarily going to be a large lumen, but relevant to the other vessels in the plant, it's going to be bigger than them.
Okay.
All right.
Okay. So the vessels, so sieve tubes.
Good.
Sieve tubes contain sieve tube platelets. So they have parts of the end walls of the sieve tube elements in the vessel, in the fluid, while in our blood vessels we don't have any platelets or end walls.
Good.
Then so sieve tube elements have companion cells present next to them, which we don't have.
Good. You're going to get 2 marks for that. I mean, this question is so broad that you could probably churn out like 100 different points, but it's a 2-mark question. Don't labor it. You get the marks there. You could talk about blood vessels being composed of tissues instead of just cells with different layers. You talk about unidirectional flow of blood through blood vessels. You could even say like veins have valves, sieve tubes don't have valves, that kind of thing. Like any sort of.
Could you talk about like specific types of blood vessel? Because like I thought like the whole. So I was about to mention that arteries and veins both consist of 3 tissues.
You would get the mark for that.
Okay.
So I appreciate your approach to just make it as broad as possible because the question is very broad.
Yeah.
But if you were struggling for answers that, compared to both the broad term blood vessel versus sieve tube, go ahead, go into the detail.
Okay. They have more mitochondria because they're very active cells. Thus they need more ATP for pumps. So they pump some mineral ions into the root hair cells.
Good, 3 marks.
Oh.
And that's it. Come on. Do you want to take another break and we'll do the other paper?
We can start though.
Yeah? Should we just go ahead? Okay, perfect. Right.
Okay. G.
So it's a bit of a trick question in that sense.
It's B.
It's not B, it's C. So the end of the hair isn't the part that's rapidly dividing, it's the bit just under to it. And that's what pushes the tip out. So here you have differentiated tissues that aren't necessarily programmed cellularly for rapid growth, whilst these ones are the ones that are programmed for rapid growth that are going to push the root hair tip out.
So is it that? So if it were D, then we couldn't take up as much water. Or plants couldn't take up as much water or mineral ions because they're not specialized cells. Okay.
When we talk about the processes in moving those water and mineral ions, that's D is where it's going to be specialized to be doing that.
It's supposed to be a bit narrower, but it's not.
It's fine. I know what you mean.
Okay.
That's good.
You could draw 2 kinetic colors.
Yeah, that's good. Go for it.
I don't know how big they are there, so that's why I didn't.
Good. So in the marking scheme, they'll give you 1 mark for centromere/kinetochores. You'll get 1 mark for the telomere, and you'll get 1 mark for saying 2 identical or 2 sister chromatids. There's one other additional part you can get by just pointing at the DNA and saying that this is DNA plus histone proteins.
Okay. So it's scripted.
Yeah. Good.
It's broken down in prophase and synthesized in telophase.
Good. You scroll down when you're ready.
Okay. Okay. So red blood cells contain cell surface membrane, which is made of phospholipid bilayer. By phospholipase breaking down the phospholipids of the phospholipid bilayer, it would destroy the cell surface membrane, which would destroy the red blood cell, and all of the contents of the red blood cell will leak into the side.
Good. You get the marks there. Whenever you see lysis, correlate it to bursting of the cell and leaking of the cell content. So you said destroy. I mean, you still get the mark.
Yeah.
And even if you didn't say that, you'd get the mark for saying the content comes out. But whenever you see the word lysis, think burst. So feel free to go back.
Okay. Similarities. Both contain no. Okay, let's start with differences. So triglyceride molecules contain 3 fatty acids,
Yep.
while the other one contains 2.
Good, that's what you want.
Similarities, they both contain glycerol.
Good. 2
marks. Okay. And so differences contains a phosphate ion group, while triglyceride don't.
Good. 3 marks.
Oh yeah. They both contain ester bonds.
Good. There you go, 4 marks. The only ones that you haven't picked up are quite I feel often with these questions you're thinking on a level of slightly deeper understanding than they expect necessarily. So you sort of pick up the marks at the bottom before you pick up the marks at the top, which is fine. You're still getting full marks, but saying something like both molecules contain carbon, hydrogen, and oxygen would probably give a mark.
Okay.
Both molecules have CO double bonds would have gotten you a mark. But I mean, everything else that you said is still so don't worry about that.
Describe the cell section or the synthesis. Oh, yeah, okay. Yeah. Smooth endoplasmic reticulum.
One mark.
Okay, describe it. So smooth endoplasmic reticulum is continuous with the nuclear envelope. It does not contain any ribosomes.
Is it continuous with the nuclear?
No.
What's it continuous with the?
Is it the rough?
Yeah, it's the rough.
Okay. So if you say something wrong, but like get other 3 marks in it, is it still?
You'll get 4 marks.
Okay. So consists of flattened membranous sacs,
Okay.
which are interconnected.
You get 1 mark for describing them as fluid-filled channels or sacs. When you say membranous.
Yeah. So the sacs are made out of membrane.
Good. Yeah. And what do those membranes do?
So they create a specific environment, so by compartmentalization.
Okay. Yeah. Good. And how do they release their substance?
By forming vesicles from the membrane itself.
Good. You get that. So if you were to say that it had a double membrane, then they would reject the answer and you wouldn't be able to get a mark for that. So you have to specify that it's just a single membrane, or just don't mention double membrane. Don't associate it with the nuclear envelope.
Yeah.
It's tubular. You have cisternae.
Mm-hmm.
You can't they don't want you to be describing it as flattened because it's real 3D shape isn't flat at all. It is a tubular structure with fluid-filled sacs.
Okay.
If you say not associated with ribosomes, you'll also get a mark. So that distinction between the smooth and rough, we are.
Okay. Oh, intercellular enzymes.
Intra or inter?
Intra.
Good. Yeah. You wouldn't get the mark you said intercellular.
Yeah. H. .5.
8.5, what were the units?
mmol per cubic
DM.
TM.
Okay, fair. Right, you just need a.
Yeah, the
Fine,
units.
that's fine. Okay. So you get the marks for that.
So wait, sorry, one question.
It's fine.
So it says show your working. Does that mean just lines?
So this is what I would do. First of all, start by saying Km equals half of Vmax, and then Vmax equals 1 AU. And then you can say at 0.5 AU, substrate concentration equals 8.5 mole, and then therefore Km equals.
Okay.
So the working part is literally just like this is how I would write it, because I want to write down every step that I'm thinking through. But the points that you're awarded for here are there, obviously, for getting the right answer with the right units, and there for just stating that Km is half of Vmax.
Okay. Okay. So the Michaelis-Menten constant provides information about the affinity of the enzyme towards the substrate.
Good, one more.
For G, the Km is smaller than that of H, thus has a higher affinity for the enzyme enzyme G has a higher affinity towards the substrate.
Good. You can, that's right. I mean, you can go on to say that enzyme G needs a smaller concentration to reach the maximum
Yeah, that's right.
velocity, or enzyme G is more likely to saturate with the substrate.
Saturate?
Yeah.
Okay. No. Okay.
So
Saturate means like a high concentration.
So saturate means that all of the active sites
Oh,
are
yeah,
filled.
Yeah.
Okay.
Yeah. Lysosomes.
Good.
Right down. So a receptor. So the cell surface membrane, the outer cell surface membrane of the mitochondria can contain a receptor which allows the lysosome to enclose the mitochondria.
Okay. Well, you're
Okay.
right, but read question D again specifically.
Okay. For the chemical signal. So, the mitochondria contains a receptor to which the chemical signal can bind to. And the mitochondria can be broken down.
So the question is asking specifically about one feature of involving the mitochondria that could lead to the release of the signals. Let's say this mitochondria
Okay,
needs to
the
be
cell has
broken down.
internal chemical messengers that signal when an old or damaged mitochondrion should be broken down. Okay. So okay. So it has something to do with the receptor, right? Or is that not correct?
I mean, at some point there will be, molecules released within the cell that will cause the cell to destroy the mitochondria, but it's not necessarily receptors that are signaling this. So if you want to think about the actual, the actual machinery involved, this might help you sort of be led to the answer. The idea that they're talking about here is that you have normal mitochondria, let's say, and for whatever reason it's damaged. And something about the structure or function of the mitochondria make these internal messengers be released, which will then activate another organelle in the cell that can destroy the...
So they can produce a signal.
So this is the signal that the
mitochondria...
Well, let's imagine that this is a lysosome.
Okay.
So it's sort of activating the lysosome that will come and destroy it.
Yeah.
And the question is asking, what's one feature involving mitochondrial structure or function that leads to this signal being released?
Okay.
It's not the best worded question.
So maybe a broken or damaged membrane.
Good. Yeah, you would get the markers like that.
Okay.
So, I mean, it could be that the mitochondria is not producing as much protein as it should.
Or ATP.
Or ATP, or, change in the distribution of its membrane proteins, or anything at all that constitutes this signal is what they're trying to get you to come to. But, yeah. Shall we find a charger?
Oh, yeah. No. I'm gonna go.
No.
Does this here? Nope.
No.
I'm looking.
Okay, sure. I'll just pause this.
What?
Let's keep going.
Synthesized by macrophages too. Okay. So first one, outline how a mutation leads to a single amino acid change. So a mutation of the GBA gene can lead to, so it leads to a different nucleotide sequence
Good.
of the gene, which leads to a different triplet code,
Good.
which leads to a different codon of the mRNA being formed,
Mm-hmm.
and thus it binding with a different tRNA and anticodon, forming a different amino acid.
Good.
Okay. In translation. And,
Thank you.
and then suggest why the two different amino acid changes result in the differences of the tertiary structure, because the R groups of so the R group of ASM is different to that of SER,
Good.
which creates different attractions between the R groups in the polypeptide,
Good.
which creates a different complex structure
Good.
of the molecule. And the same for, well, not the same, but so it also causes a change in the structure
Mm-hmm.
of the R group system.
Yeah. There's one more piece of information at the table that you want to use in your answer.
Which can cause more severe
disease, yeah.
than other.
And you suggest to me why.
Because the change in the tertiary structure is bigger.
Yeah, okay. And what are we actually talking about? What is this protein?
It's synthesized by macrophages.
Mm-hmm.
And lysis a glycolipid.
Yeah, cool. It's an enzyme, basically. Okay. And so we're mutating an enzyme here.
Yeah.
One mutation gives a mild disease. Another mutation gives a severe disease. Can you guess what these mutations are affecting?
They can affect the active site, the shape of the active site.
Good. So in the leucine to proline mutation, you might have more of a disruption to the active site, which disrupts the function of the enzyme further. Good.
Yeah.
So that's a huge 5-mark question, but you'd get marks for that. The only paper where they don't ask you to name it, but
Okay. Okay. So a loss of chlorine ions and sodium ions will cause the solute potential of the environment to increase, thus decreasing the water potential of the environment. And
which environment?
So of the well, the environment of the small intestine.
Good, the intracellular environment.
Okay, the intracellular environment. And so thus creating a concentration gradient.
Good.
For the water to diffuse down, or move to biosmosis.
Good.
Okay.
So movement of ions sets up an a water potential,
Yeah.
which allows water to move by osmosis from a high concentration to a low concentration. Good.
Okay, so outline the transport mechanisms that result in the movement of glucose and sodium ions across the intestinal epithelial cell. So for sodium ions, they're actively transported and pumped through carrier protein. So they're pumped into the cell.
Good.
Okay. And yeah, glucose. Okay. Glucose moves through a co-transporter protein by facilitated diffusion.
Okay.
Good to concentration gradient.
And why is it called a co-transport?
Because it moves together with sodium ions. So glucose moves together with an ion.
So what I'm trying to get at is both the sodium and glucose are being co-transported by facilitated diffusion. Okay, we'll get 2 marks. Okay.
And then the other suggests how movement of glucose and sodium ions increases the water uptake by the cell. So it decreases the water potential inside the cell, thus setting up a water potential gradient which allows water to flow into the cell by osmosis.
Yeah.
From high concentration to low concentration.
Okay, good. The only thing you need to add to your response there is the transport mechanisms beyond just coming in from the lumen and into the cell. So they want you to talk about how it goes then from the cell to the capillary as well. So in the first part of the question, when you told me how sodium and glucose enters the cell, you also have to talk about how sodium and glucose then exit the cell to go into the capillary. Does that make sense? So use that diagram and just add that part to your explanation.
So, okay. So glucose is transported out of the cell by a pump by active transport out of the cell into the
Is it active transport?
No.
No, if you don't know for a fact that it's going to be that it's active transport because you see that ATP being used, then you can assume that it's going to be facilitated.
Okay. Yeah.
And how about the other ion?
So that would be sodium ions.
So we've gone to the stage that sodium is being co-transported into the cell with glucose.
Yeah.
Now tell me what happens to that sodium.
So that sodium can be transported out of the cells by a sodium potassium pump.
Okay. So now we've gone to the stage where we've described glucose and sodium entering the cell from the lumen of the intestine, and then both exiting the cell into the capillary. And now that you have that picture, you can talk about how the movement of those ions are moving water alongside.
Yeah.
So by moving ion from lumen to cell to capillary, you're setting up a water potential that causes water to follow the movement of ions.
Yeah.
And that's how we rehydrate these patients.
Okay. Yeah.
Makes sense? Okay, let's move on.
Right. Yeah. Arteries contain blood with a high blood pressure.
So
if so they would a lot of blood would be lost if you were to hydrate it that way and inject and damage the vessel.
yeah, so if you were to basically screw up the insertion of the cannula, then there's a higher risk if you're going for an artery because the risk of blood loss is greater. The pressure part, it makes it more difficult for the drip to stay in place because you've constantly got this pressure like hitting against the drip.
Okay.
Which is why we'll put it in a vein instead. And you can also mention that veins are easier to reach than arteries because arteries tend to be deeper.
Yeah.
Okay, that's good. You get them all?
Various cases. So it will inhibit or prohibit translation from occurring.
Okay, so what does that mean?
It won't be able to form the proteins needed for it to grow or
Okay,
survive.
so they're asking specifically about the effects on metabolism.
Okay. Yeah, so it won't form the enzymes to catalyze metabolism.
Good. Metabolic reactions. Yeah, you get them all. I want you to do this like simply, just try and do it as systematically as possible. Start with the really broad, obvious stuff.
Okay. So all curves are decreasing
Good, yeah. So over time,
over time. Okay.
diarrhea is reduced to 0.
Yeah.
Regardless of the treatment group. That's 1 mark.
Okay. So diarrhea is reduced. Well, it's not. Oh, it's still okay. Diarrhea is reduced to 0 at 64 hours for both the multiple dose and the 1 dose of 1 gram.
Is it? Double check.
Maybe not.
Yeah, look at the lines.
Oh no, it's wait. Multiple dose, and isn't it?
So multiple doses the line with the dots. So it's
Yeah,
a broken line
so-
with dots.
Yeah,
So that one's going down to 0.
and the 1 dose.
Good. Yeah. Okay. So what are you saying? What's the comparison you're
Okay.
making?
While the one without antibiotic and the one with 2 grams is reduced at 128 hours to 0.
Yeah. The others are reduced at 64 hours. Okay. Good. That's another mark.
Okay. Trend. Then, okay, the one without an antibiotic causes the patients to have more diarrhea from most of the 128 hours than the others.
Yeah.
And the one with 1 gram of whatever, the antibiotic causes the patients to have the least amount of diarrhea. Right?
Yeah. You've made enough comparisons here to pick up the marks.
But they were
That
like
was
more
good.
simple ones, so.
No, I think you actually made them all the relevant ones there. You definitely get better at it. I mean, definitely start with the broadest terms. Regardless of treatment, diarrhea reduces over time.
Mhm.
I would then say compared to no treatment, all of the antibiotic groups, all the antibiotic treated groups had a greater reduction in diarrhea over the time period tested.
Okay.
And then go on to say the 2 gram dose of tetracycline only ended the diarrhea at 128 hours, which was the same as the no antibiotic group. On the other hand, 1 gram or continuous dose finished at 64 hours.
Okay.
That kind of thing. I mean, you will get the marks for it. There are only 3 marks to pick up, but just try and be as thorough as possible.
Okay. So they do support using tetracycline because so patients have overall less diarrhea than if the antibiotic wasn't used. And with 1 gram and I don't know the other one, but with more
We'll just call it the multiple dose.
okay, multiple dose. the point of the patients having no diarrhea is reached twice as fast.
Good, you pick up the first one. And now if I told you ARC, you do not support the treatment.
The one with 2 grams takes as much time to set the diarrhea to 0 level as the one with that antibiotics.
Yeah, and also in all patients, it resolves. And if I asked you to argue why you're not able to say whether or not the results support it.
Because they are not enough people tested.
Yeah, that's a valid point.
Because the 2 gram antibiotic behaves different, right, to the 1 gram of the multiple dose.
Yeah. How about the idea of measuring diarrhea as measuring the volume of diarrhea when you're going to treating?
It's not the only factor.
Yeah, I mean, okay, diarrhea is one thing.
Yeah,
but
it might be something.
It's death that we're trying to prevent, right? So, yeah, you want to like measure, make sure that what you're measuring correlates to like the clinical outcome you're looking for.
So if you said you don't like that it, so you could say that it does support or it doesn't support.
Yeah, and you can put arguments for both as well. Like if I was you, always stay on the safe side.
Yeah.
Hey, why not? Okay.
Okay, so they don't want to increase the resistance of Vibrio cholerae towards this antibiotic. So by giving it multiple doses of the antibiotic, we increase the chance for the mutation to occur.
Do we increase the chance of the mutation to occur?
No, we don't.
Not necessarily.
We
Okay, I'll try and talk through it in a sort of systematic way. So like moving away from the biological mechanisms that are going on here. This is a really broad question. It's saying it's antibiotic resistant. Explain why this means that medical practitioners prefer to treat cholera with a single dose of antibiotic. You pick up the marks by just saying that it's more likely that a patient would take a single dose. It's easier to deliver a single dose. So that would resolve the infection more readily. It doesn't directly answer the question, but you would pick up a really simple mark there. And then when you come to talk about bacterial resistance, always think about it in the perspective of bacteria that aren't resistant, so bacteria that are susceptible, and bacteria that are already resistant. So if you have a group of bacteria that are susceptible to tetracycline, and you give them 1 dose, we have a chance of wiping them all out with 1 dose that's big enough to kill them. And that's better than wiping out some of them with multiple doses, because then you're leaving time for the bacteria to basically divide under natural selection. So it's not that giving less antibiotic increases the chance of mutation, it's more that it then gives a selective advantage to those bacteria that have the mutation.
Okay.
So they will proliferate further, and then you're basically culturing antibiotic resistant species.
Okay.
Okay. And then, I mean, you'll get more marks as well if you go on to say that having antibiotic resistant species is bad, because then you'll have patients who essentially deposit those into the environment and make the global epidemic worse. Just finish on these couple, then we can pick up the rest next week.
Most people are very So the antibodies all contain different variable regions of antigen receptors, which are complementary to the specific proteins of the toxin, which are different to the specific proteins of the Xylem, which are also different to the specific proteins of the main bacterial cell.
Okay, so you get 2 marks there. 1 mark for saying that you've had complementary antibodies. Another mark for saying that those antibodies are complementary to the different things.
Mm-hmm.
To your last mark, they want you to focus on explain why the antibodies are different.
Okay. Because the B cells that attach to the toxin main bacterial cell, the bacterial flagellum, are all different, and they form, so they have different receptors for the antigens. Thus, they have different DNA which codes for different antibodies.
Okay, so I mean, the way that actually works, if you want to go to hierarchy, it's that you've put these 3 different antigens into the serum. Each one has its own specific epitope. You already went through that, so you get the mark for that. Then each one will activate a different specific B lymphocyte.
Yeah.
Which will then go into plasma cell and clonally divide.
Yeah.
And secrete antibodies with different variable regions that are specific to each of those epitopes. So you got all the points, but you just you want to structure it in the sort of sequence of how it's happening. But that's good. Okay, let's start there. We can pick up next week.